Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For this section to discuss NCERT Class 12th molecular basis of inheritance, that is mutation and genetic code. What is that mutation? What is that in a genetic code? See, the relationship between genes and DNA are the best understood by mutation studies. Here, mostly the relationship between genes and DNA. Genes and DNA. Already you know that for example, you take as a one particular the DNA. Right? This is the DNA. That DNA they contain as what? The DNA contain as a genetic information is present. Okay. So the DNA and genes, their best understanding for as an A, the mutation studies. You have studied about as a mutation and then the effect of in chapter 4. So in the principles of inheritance, I thought in the principles of inheritance in the chapter we clearly discuss about as in this enter the mutation. Okay. And effects of the large deletion and rearrangements of in the segment of the DNA is easy to as in a compiled. So in that, for example, here AUG is present. Here they having as what? AUG is present. So if in case you get add with as an cytosine, that is here mutation takes place or not? Mostly here the mutation will be takes place because they having only single base pair is present. They having is what? Only for the as in a single base pair is present like. And then let's see here. It may be the result in loss or gain of any genes in a function. So in the total DNA stand, any codons will get add or any codons will be their loss. There what happened? Almost their mutation will be takes place. For example, here they given as a name Rom has big cat. Rom has big cat. Here they have 3 codons is present. Here they having what? 3 codons as present. So in these 3 codons, at least you get remove here one by one. If in case first you remove as an B, after you get remove as an I, after you get remove as an G, then what happen? Mutation is takes place. Mutation is takes place. They given as an that one also clearly. Let's we see here. Now the effect of the point mutation will be there explained here. They given like. A classical example of point mutation, the change in a single base pair of a gene in a beta globin chain result as in a change the amino acid, the residual guanine to as in a alanine. Very, very, very important. Here point mutation means it is a total DNA, right? In this total DNA, only one single base pair. See, it is only single base pair will be get changed. Their single base pair will be get changed. Then what happened? That is the total DNA, the sequence will get changed. That is called as in a point mutation. In the point mutation, we see that in a see here glutamate is converted into as in a valine. Glutamate is converted as in a completely valine. See mostly in that it result as a completely disease condition is called as a sickle cell anemia. So the time they get disease is what? There is a sickle cell anemia. The effect of the mutation that is in a insert or deletes as in a base structure genes can be that better understood for that given as in a simple example. So they given as the simple example also given once we see here that one. Okay now, like next. The concern statement that is made up of as in a following words, each they having a 3 letters like as in a genetic code. See first has in a in that one Rom has red cap. Rom has red cap. Am I right? So before in this codons we are adding with as what? Big B-I-G. Let's see, we are the insert the letter B between the has and red. It is a rearrangement of the statement would be the red like. See, Rom has. Now between has and red we are adding with as what? B. Am I right? So when we are add with as in B, what we getting? B-R-E and D-C-A. Here P only is there. Am I right? This P is called as what? In this P they get as a mutation. It causes as what? In a mutation. Let us similarly we would they are completely now insert as in a 2 letters in the same place in a BI now would be read. Now we are add with as BI. Now we are add with as what? BI. Now you getting as what? BIR, C, DE, DC and AP. Here they are having a 2 their base pairs as present. Here also takes place as what? Here also takes place as a mutation. Here also takes place as what? Mutation. And now insert the 3 letters together like as a BIG. Now these three letters we are get and what happened? Ram has big red cap. Here, the same in this process here of what happened that they have three nucleotides as present. Yes, na? Here the three. So here mutation is takes place. No, because here they having as in three codons as present. So that purpose here mutation is not takes place. Now it is a what? It is addition of in a codons. Here addition of the codons. Let's see here after the deletion of the codons. Let now we discuss here the deletion. See, in the same exercise, it is repeated the deletion of in a R E D one by one in the rearrangement of the statement make as a triplet word. See here now one by one to get remove. So first what first you B I G. So first you remove as in that completely G then B and then B. Here deletion process. ROM has EDCAP. Here when we are removing as word G. Here now we remove as an IG. That is what now gives ROM has DCAP. Now again here remove as in B also finally get as what? ROM has cap. So when you completely we remove as an BIG, when you remove as an BIG, finally we getting as what? ROM has cap. Let us. The conclusion from above the exercise is very there obvious. The insertion or deletion of any one or two the bases change completely reading with as from as in a point insertion or in a deletion. Mostly any that in a the codons to get inserted or any to get deleted. Okay, let's. However, such mutation is referred to as in a that is frame shift mutation or deletion mutation. That is a frame shift insertion or deletion mutation. That is what insertion or deletion of three or its multiple bases insertion or deleted in a one or multiple codons in a one or in a multiple amino acids. That is called. It is regarding as a frame remain as in a unrelated form of in a point of choice. So it is called completely in a frame shift mutation. Okay. Let's we discuss tRNA, the adapter molecule. For the very beginning of the in a proportion of codon, it was clear the Francis Crick that there was to be as a mechanism to read the codons. Also, they link the amino acids because the amino acids having the no structural specialized to the read the code the uniquely. So here it is then completely. It is a part of the mRNA strand. This is what it is completely in mRNA strand. So this mRNA strand to get completely see here mostly in this mRNA strand to continuously to get the rate and in this amino acids to get bind with as in that in a specific sites. They bind with as in a specific sites and this postulate the presence of the adapter molecule that would be their one hand for read codons and other hand would be bind as in specific amino acid. So here one hand to get read and one hand will be to get bind. Okay, and the tRNA is called as in the soluble RNA. The symbol called as an sRNA. It was before their given genetic code was in a postulated. So here the sRNA that soluble RNA it is the completely where genetic code there was postulated. However, its role of the adapter molecule was assigned as a much later. So get major function is what it is get act as an adapter molecule. Okay, let us here the tRNA has an anticodon loop that was with the basis of complementary to as a code for as a what amino acid one acceptor N2 which has an bind with as an amino acids in a tRNA specific amino acid. The specific amino acid the initiation is takes place. So here the tRNA once you notice here this is in a tRNA. This is the tRNA stand is there here. It is in a tRNA stand. It is an anticodon loop. Loop means one bunch. It is what one completely anticodon loop. It is complemented to as an codons to a reader. So it is their binder. It is the reader. Yes, na mRNA is reading stand. tRNA is to get binding stand. Which bind with as an amino acid. The tRNA the specific of amino acid. For the initiation, there are in another specific tRNA that will be refers to as an initiator tRNA. So first when they get bind here in this region, that entire the codons is called as what? Initiator of tRNA. These are no the tRNA stop there. Sorry, in this no tRNA for stop codons. They have no stop codons for in this process. Here they given as in figure 5.2. So they given as in here they have no stop codons will they get given. Let us the secondary structure of the tRNA has been there depicted to as in look like as in the clover leaf or actual structure of the tRNA they combined with as a molecule look like a inverted L. So when you are there complete to get appear like as an inverted L shape or as in the clover leaf shape also there appear as in this tRNA molecule. Okay, this is in the adapter molecule, the tRNA adapter molecule. Okay, this is next to let us we discuss translation. What is a translation? Next one, what is that? Translation. Translation to refer to as a process of polymerization of amino acid to polymer of in a polypeptide chain. Here, the process of polymerization of amino acid to form as any polypeptide chain, that is called as any translation process. Now, in the order sequence of amino acids is defined by as a specific basis of mRNA. So here one code will be to get complete they get move like, and another codon they complete their codons will be to get bind as in the specific mRNA stand. And so for example here they given it is in mRNA stand, right? It is in a one part of the mRNA stand. So here they complete the different amino acids to get bind here. Here the different here amino acids to get bind, like to get bind with as an the specific amino acids for this region. Let us the amino acids are the joined by as bond with as known as the peptide bonds. When they are complete the amino acid they comes here like see the serine, proline, arginine, like in these the amino acids to get bind the specific amino the specific bond is present. That bond is called as what? That is in a peptide bond. That is in a peptide bond will be there present. The formation of peptide bonds required the energy. They require as in what? Energy they require means ATP is required. Therefore, the first phase of the itself amino acids they activated the presence of the ATP, the link to as in a cognate tRNA process commonly called as a charging of tRNA or amino acylation of tRNA. So when they get using as an ATPs, this process is called as what? Amino acylation process. Amino acylation of tRNA or called as in what? That is in charging of tRNA. Charging of tRNA. And then after here to be the more specific that two such as charged tRNA brought close to enough their formation of the peptide bonds between them as in would be they are favored in a energetically. So they are completely form as how that they form as in mostly they are energetically. Let's see here. These are the amino acids. These are the amino acids. They are bind with as in with help of what? It is in a peptide bond. This is what? Peptide bond. They require energy. They are taking from here. Let us. The presence of the catalyst would be there. Enhance the rate of the peptide bonds will be formed. So when they are complete, this code will be they get read, and automatically the tRNA will be get bind and to get form as an amino acids. In these amino acids, they get forms as an A with help of peptide bonds. They are complete the chain-like structure. The cellular factor responsible for as a synthesis of proteins, that is the ribosomes. So here these are the ribosomes. These are the ribosomes. It is a small subunit. It is in a large subunit. It is in a large subunit. Already you know that in a prokaryotes has in a 70s type of ribosomes and eukaryotes has in 80s type of ribosome is present. Let us the ribosome consists of as in structural RNA is about as 80 different types of protein. Here they completely they contain as how many types? 80 different types of the proteins will be there present. Now inactive the state the exists as a two subunits, the large and small subunits. So this is a small, this is a large. The small subunit encoded as an mRNA. So in the small this is a small subunit. Then encoded by as what? mRNA. 2. The protein to begins there are two subunits of the large subunit. There subsequently the amino acids to bind with as in this that closely end up the each as in the formation of any peptide bond. So when this the large subunit in the tRNA will be the cognate, then what happen? It is to get forms as a number of amino acids will be the get form number of amino acids to get forms. Okay. Next here the ribosomes also get act as a catalyst. See rRNA, that is 23S rRNA, the bacteria, the enzyme, the ribosome enzyme, their formation of peptide bonds. So here rRNA also it is to get participate in the formation of any peptide bonds. Okay. Next, the translation unit, the mRNA is a sequence of RNA. They are flanked by as in start codon, that is in a AUG, and the stop codon, that is the code for as any polypeptide. So here one codon will be there, initiator codon is there, that is the AUG, am I right? So initiator codon is there, that is the AUG. They start that as in codons, and the stop codons also is there present, like as in UAA and UGA and UAG. Yes, na? These are completely what the stop codons also they are present. When the stop codon is present, what happened? They are completely stopped with as the formation of peptide bond. The mRNA also gets some additional the sequence that has no translated region that refers to as an untranslated regions. So where that information to get not pass, that regions is called as what? UTR regions. That is called as what? Untranslated regions. The UTR as presence of the F5 and before the start codon, the 3 and of after the stop codons, they are required for the efficient the translation process. Very very important this point. See, mostly the stop codon function is what? They are required for the efficient translation process. Efficient translation process. These are very very important for as any the stop codons. Okay, this is next. Next, for initiation, the some ribosomes are bind with as an mRNA. They start codon AUG and recognized as only one by the initiator with the tRNA. So here this codon how to get bind? This mostly this codon. For example, this is an mRNA stand, right? This is in mostly in a mRNA stand. This mRNA stand to get bind with as in specific amino acids. They are bind with as some specific amino acids. Their code will be start here in the presence of the codon is what? AUG. That is what? AUG. Here when the AUG will be there bind, what happened? The ribosomes they are completely the proceed as an elongation phase, the protein synthesis. That during the stage the completely complexes composed as an amino acids linked with as an tRNA. Then what happened? Subsequently they bind with as an their appropriate codon mRNA forming as an base pair to as an anticodon. So it's a codon, it is an anticodon. They come to get bind, they get formation of what? They come to get formation of any ATP molecule. Let us the ribosomes move from as a codon to codon along with any mRNA. So here the enter the codons will be continuously to get formed. The amino acids are added one by one. The translated the polypeptide sequence they're completely detected by as a DNA presence of any mRNA. So mostly here the information to get comes here the synthesizing and information to get completely after when the synthesizing information to get moved like as an amino acid with the help of the polypeptide chain. It is this end of the release factor. One is in initiator factor is there. That is the next one is what? When the releasing, that is a releasing factor to get stop codons will be there present. In the termination, the translation is releasing as in a complete the polypeptide chain form will be there form from as in a ribosome. So when the complete the releasing factor will be the get form in the presence of in what? In a translation region of the polypeptide form as in a ribosomes. Okay, now discuss in next class. Thank you for watching. Please share, subscribe and like.